2020. The coronavirus is bad, but there might be a solution on the way. Vaccines. But how is this vaccine delivered so fast? How does it differ from other vaccines I may have heard of? But most importantly, how does it use these new mRNA tactics to combat COVID-19? All this and more in our Group 4 project! But before we get to all that, we have to understand, what are vaccines? Are they healthy? Are they a government microchip implanted by Bill Gates to keep you subservient?
Wake up, sheeple!
Great question. The function of a vaccine is to stimulate an immune response specific to a certain pathogen in order to equip the body with the cells it needs to combat the pathogen in future. Typical vaccines do this by injecting a weakened or dead form of the pathogen or its antigens into the body. An antigen is a part of a virus that an antibody binds to during an immune response. In the COVID-19 virus, these are club-shaped spike glycoproteins. The antigens are specific to each species of virus, meaning that each species has a unique set of antigens, and instead of being able to launch a general immune response, each time you're infected with the virus, your immune cells have to find the correct antibodies to launch the specific immune response for that virus. However, recent technology has allowed scientists to experiment with mRNA vaccines, which generate a stronger immunity by stimulating the immune system to make both antibodies and killer T cells.
Professor, how does mRNA factor into all of this?
To start with, the genetic code for all the proteins in the body are stored in DNA and expressed with the help of RNA, which are molecules made from long chains of nucleotides, each representing one of the 4 complementary paired bases. DNA is always kept in the nucleus of the cell, and therefore cannot be decoded directly into proteins, because the nuclear envelope separates it from molecules called ribosomes, which build the protein. So messenger RNA, also called mRNA, is used to carry a copy of the gene sequence in the DNA to the ribosomes for it to decode into proteins. In a process called transcription, RNA polymerase binds to a section of DNA, which marks the beginning of a gene sequence. Enzymes unwind the DNA helix, exposing the strands. RNA polymerase uses a strand of DNA as a template to create an mRNA strand with bases complementary to the DNA.
But how does...
The first thing to understand is that a protein is just a chain of smaller monomers called amino acids, which are made into proteins during translation. Initially, the ribosome binds to one of the mRNA, then the ribosome slides along the mRNA to read it. Each set of 3 nucleotides, called a codon, corresponds to one of the 20 amino acids found in the human body, and this codon binds to another type of nucleic acid called tRNA with complementary anticodons and the corresponding amino acid. The amino acid on each tRNA molecule forms peptide bonds with its adjacent amino acid, forming a polypeptide, which folds up into a 3D structure and then carries out its function in the body once it's released.
I'm starting to understand. But how does any of this fit into vaccines?
mRNA vaccines operate in a similar way to classic vaccines, except instead of injecting antigens, they give the body instructions to produce them. The mRNA sequence that codes for the SARS-CoV-2 spike proteins is injected into the upper arm, and dendritic cells that can display antigens on their surface, in the muscles use ribosomes to read the mRNA and assemble amino acids into spike proteins, which are either displayed on the cell surface or expelled out of the cell. The spike proteins can trigger an immune response, but are not harmful at all. The mRNA strand is then disposed of by enzymes in the cell, such as ribonuclease, just as in normal translation.
So I'm starting to understand the theory. You take the mRNA, put it into the dendritic cell, it gets transcribed and translated into spike proteins, which are displayed on the outside of the dendritic cell, and then the immune system takes it from there. But how do we get the mRNA into the cell in the first place?
RNA is a notoriously fragile molecule and is not designed to be transported in and out of cells. So the molecule is encapsulated in a lipid nanoparticle to ensure that it passes through the cell membrane without being broken down by ribonucleases before reaching the target cells. In the Moderna vaccine, this lipid nanoparticle is made from 4 different lipids which bind to the mRNA, prevent it from being destroyed, and stabilize the structure. The phosphate groups in mRNA are negatively charged and interact with the positively charged ionizable lipid to create a lipid nanoparticle structure. This structure, similar to the cell membrane, is made from a lipid bilayer with hydrophilic parts facing outwards and hydrophobic parts facing inwards. This particle enters the cell through a process called endocytosis, causing an additional layer of cell membrane to surround the particle. The cationic or positively charged lipids from the nanoparticle interact with the anionic or negatively charged particles from the cell membrane, creating conical structures instead of the usual flat bilayer, causing a disruption in the arrangement of the lipids and releasing the mRNA into the cytoplasm where it is able to be translated into spike proteins.
Get out of here, Vara. I'm the only green screen guy in this town. But that still leaves one question. The mRNA is inside, it's been transcribed, been translated, the spike proteins are on the outside of the cell, but how does that cure the virus?
What happens after the body makes SARS-CoV-2 spike proteins? Killer T cells, a type of lymphocyte, recognise the cells that display the spike proteins on their surface and destroy them. And helper T cells recognise the expelled protein spikes and signal B cells to create antibodies specific to the spike proteins. The cells involved in this immune response differentiate into T memory cells that remain in the body, so if the virus infects you a second time, these T memory cells can rapidly create the correct B cells, launching a specific and effective immune response. The benefit of this process is that both B cells and T cells are triggered to respond. B cells from the expelled spike proteins, and T cells from the fragments of spike protein displayed on cells, which does not happen when we simply inject the pathogen like in normal vaccines, which means mRNA vaccines can actively produce a much more complete immune response.
One question. Are you telling me that we can just synthesize mRNA? Based on spike protein data that we have? How?
mRNA vaccines can be developed in a lab using a DNA template. This means the process can be standardized and scaled up, making vaccine development faster than traditional methods. To make the mRNA, a DNA template, usually in the form of a plasmid that is complementary to the desired RNA, is transcribed by bacteriophage RNA polymerase, an enzyme responsible for transcription in certain viruses. The 5' cap and poly-A tail can be added enzymatically by enzymes such as poly-A polymerase. The mRNA is purified through precipitation and extraction, and all the DNA that remains in the mixture is then digested by the DNA enzyme. Substances that help trigger immune responses, called adjuvants, are then added to increase the vaccine's efficiency.
Sounds a bit hard. What kind of technology do we need to do this?
The coronavirus RNA is currently undergoing a whole genome sequencing using a variety of technologies, mostly next-generation sequencing. Next-generation sequencing, also known as massive parallel sequencing, is distinct from older sequencing methods as it can analyze anywhere from 1 million to 43 billion strands of DNA, which can be up to 400 bases long, simultaneously. The most popular of these techniques is that developed by Illumina, which is estimated to be used to produce 90% of the world's sequencing data. While it relies on the same basic principles as older sequencing methods, in which DNA is broken into fragments, copied to create templates, and then these are used to recreate the strands using fluorescently tagged nucleotide bases, it also incorporates a number of innovative techniques as well as a general improvement in technology to allow for faster and cheaper genome sequencing on a much larger scale. This technology means that the genomes of new variants can be quickly sequenced and a new vaccine can be created just by modifying the starting DNA template.
You've convinced me.